Hi guys, Miss Francis here. Today I'd like to start our discussion on cell membranes. What's super important to remember about cell membranes is that they are selectively permeable. They determine what comes in and what comes out. So it provides a barrier between the inside of the cell and the outside of the cell, where the main macromolecules in the membranes are lipids and proteins, where the most abundant lipid is the phospholipid. And if you remember from our biochem unit, phospholipids are amphipathic, meaning they have both hydrophilic and hydrophobic regions. So the membrane is fluid. It has proteins embedded or attached to a double layer of phospholipids, a phospholipid bilayer. And it's a fluid structure that's dynamic. It isn't static. We didn't always think that the membrane was this fluid phospholipid bilayer. Over time, scientists made different observations that helped us infer what the actual shape of the membrane is. So the first observation that gave us some sort of clue as to what the cell membrane might look like is that Charles Overton noticed that substances that dissolve in lipids enter cells faster than those that are insoluble, that don't dissolve in lipids. So he hypothesized, well, then membranes must be made of lipids. Then there was a chemical analysis of red blood cell membranes, and it showed that the membrane contained lipids and proteins. So by 1915, we knew that lipids and proteins were the components of cell membranes. In 1925, we discovered that the cell membrane is made up of amphipathic phospholipids, where the cell membrane must be a phospholipid bilayer that's 2 molecules thick, because water solutions both fill and surround cells. Actual membranes, though, adhere more strongly to water than do artificial membranes that are composed only of phospholipids, which led scientists to the inference that there are proteins that are on the surface of the membranes that increase adhesion. So at this point, if we were to identify which of the following diagrams accurately represents a phospholipid bilayer in our cells, we would be looking at the arrangement presented in option D, because it shows the hydrophobic fatty tails that are sheltered from the water, while the hydrophilic phosphate groups are interacting with the water. So now, adding the knowledge that proteins on the surface of a membrane increase adhesion, scientists Davson and Danielli proposed a sandwich model in which the phospholipid bilayer lays between 2 layers of globular proteins. So the globular proteins rest on top of that phospholipid bilayer. However, 2 observations that were made were not consistent with the davson-danielli model, which was, first of all, not all membranes were alike. They differed in thickness, they differed in their appearance when they stained, and there was a different percentage or ratio of proteins to lipids in each different membrane. Furthermore, membrane proteins are actually not very soluble in water. Instead, they're amphipathic with both hydrophobic and hydrophilic regions. So with the davson-danielli model, they show the hydrophobic regions in contact with the water, and that really just doesn't make any sense. So what we have come to is this present fluid mosaic model of the cell membrane that was proposed by Singer and Nicholson. Now, mosaic means that it is speckled. It has a lot of different shapes and sizes and colors. And that's what we've come to believe is what the cell membrane looks like, where the hydrophilic regions of the proteins and phospholipids are in maximum contact with the water, whereas the hydrophobic regions are in the non-aqueous environment. And how did we figure this out? Well, the electron microscope helped us find new ways to study the cell membrane. Remember, we originally had the light microscope, then we had these electron microscopes, which form an image by shooting the electrons through an object. And by 1944, the electron microscope provided us with 2 nanometers of resolution. A freeze fracture is the technique that's known as what showed us how the proteins are speckled through the phospholipid bilayer. So freeze fracture, it splits the membrane along the middle of that phospholipid bilayer, and by examining the pictures of what was seen, you could see that the inside of the cytoplasmic layer has these proteins, these bumps that we see in the picture. Again, here's another picture showing the inside versus the extracellular surface of the membrane, where inside there are many bumps, which we infer to be those proteins. Again, analysis of the falsification of the davson-danielli model led to the Singer-Nicholson model, which is the fluid mosaic model that today we believe to be true, because the fracture reveals an irregular rough surface inside that phospholipid bilayer,